My name is John Cohen. I'm going to moderate the session, and I'm also going to open with an overview of CRISPR. Two of our scheduled panelists didn't make it, so there'll just be the three of us. But the upside of that is that we really want to have a more interactive discussion with all of you. So please think about questions, ask questions, even be provocative. These two guys have a lot of knowledge. I cover CRISPR, and I'm going to start with an overview of CRISPR. I hope that I'm not speaking down to anyone. I do find that a lot of journalists who write about CRISPR don't really understand the mechanics all that well. So I'm just going to start with, do I need to click on this? Yes? I guess I do. Can you load the program CRISPR 123? Or can I load it? I don't see it there.
You would like your presentation.
Oh.
You would like your presentation?
Yeah, mine is, oh, I see. Mine's my name there, the bottom one. Yeah, thank you.
That's perfect.
Yeah. So when I first became involved with reporting on CRISPR, it looked to me like an electrical wiring diagram. I thought it was mind-boggling. And I studied this CRISPR illustration on the right for many, many hours, and then I went to the Cold Spring Harbor Laboratory CRISPR meeting, which is the CRISPR meeting. I was the only journalist there. And I sat at the bar with Jennifer Doudna and a group of other pioneering CRISPR researchers and told them that no one could make any sense of this diagram, that this required a special degree in CRISPR to decipher it. And I made fun of them for being such terrible communicators, and they made fun of me for being so stupid. This isn't my next slide, so I'm way out of whack here. Hang on. I don't know why it's jumping around here, but let me back up. Ignore what you're going to see for a moment. Close your eyes. So this is the CRISPR illustration that most people see, that many journalists use to illustrate their stories. And to me, when I saw this, when I didn't really understand CRISPR, I thought of the old lady in the shoe. That's what it looks like to me. It still doesn't really tell me what's going on. And then when I got down very deep into the mud and really started to decipher what CRISPR was, I realized that it was incredibly simple. All it really is guiding the CRISPR mechanism to a target. Then CRISPR cuts the double-stranded DNA, and that's it. The cell repair mechanism is everything else. All CRISPR does is cut double-stranded DNA. That's what vanilla CRISPR does. It doesn't do anything else. All that complicated stuff, all the electrical wiring diagrams, the old lady in the shoe, all of that is extra detail. I know what that extra detail means. It helps me write about CRISPR, but this is all I really need to know, and this is all I really think journalists need to fully grasp to understand when they write about CRISPR, that after the DNA gets cut, the cell repair mechanism does everything, and it goes down one of two pathways. One pathway is it knocks out the gene, it cripples it, and the other is it allows you to knock in something or add something to the gene. CRISPR also comes in different flavors that aren't CRISPR. CRISPR has predecessors. So you sometimes may hear about TALEN or zinc fingers or meganucleases. They do the same exact thing. They cut the DNA, and then the cell repair mechanism takes it from there. The complicated stuff, the two arrows that I had in the earlier slide of the knock-in and the knockout, the complicated stuff is non-homologous end joining or homology-directed repair. Those are the complicated things. And I hope that the researchers who do CRISPR can explain that to you simply. I just want to quickly run through what the promise is of CRISPR. Part of the promise is in agriculture. The plant in the middle here, these are wheat plants. has powdery mildew, which is a fungus that ruins a tremendous number of crops. The two plants on the outside are CRISPRed to resist that, and even though they're sitting right next to that plant, you can see that they look different because the powdery mildew cannot do anything to them. Wheat is really weird. Wheat has 3 combined genomes. It's 17 gigabases. Humans are 3.2. So wheat is enormous. It's really hard to change all these genes. CRISPR can change several genes at once. There is an application of CRISPR in animal that runs in 2 directions. One is to do livestock changes, and another is to create disease models. And the third is to do transplantation of animal organs. Gene drives are another application of CRISPR. Essentially, the simplest way I think to understand gene drives is it puts the CRISPR mechanism into the chromosome, into the gene, and it passes on from generation to generation so that you can more quickly make a gene travel through the tree in reproduction, and in this case, trying to get rid of, let's say, female mosquitoes. You can do it in a few generations because CRISPR is being inherited along with the gene and making the cut or changes or additions that you want to make. CRISPR changes break into 2 categories. One's ex vivo, one's in vivo. The ex vivo will pull blood from people, make a change in the blood, put it back in. In vivo will put the CRISPR mechanism inside the body and let it make the change in the body. And then there's germline editing, which has received far more attention than anything else CRISPR related and is one of the most fascinating aspects to it. It's also the least advanced, and there have only been, by my count, 12 or 13 human embryo experiments with CRISPR. So very few people in the world have even played with this, with human embryos. Finally, I was told by Rudy Janisch, who did the first complicated mouse with CRISPR, where his group modified several genes at once. I was told by Rudy Janisch that any idiot could do CRISPR, so I decided to try. And I did a video about this for Science, and I did it side by side with a bona fide CRISPR researcher, and I failed. It just underscores to me that when scientists talk about how simple something is, it doesn't really mean it's simple. I don't know how to pipette. I've never worked in a lab. I've been a science journalist for 30 years. I'm a really bad pipetter, and I blew it. Someone pointed out to me that meant I'm not an idiot, which I took as a half of a compliment. But at this point, anyway, we're going to talk to some scientists who are not idiots who do this and succeed at doing it. We'll hear from Chris Proudfoot, who works at the Roslin Institute and does CRISPR work with animals, and Jean-Denis Faure, who works at the Institute, the National Institute for Agronomics Research in France, and does plant research. So why don't we start off with Jean-Denis. I think I offended him earlier when I suggested we go that direction because plants are not as evolved as animals, but we'll see what you have to say. Thank you.
Thank you very much for the introduction. So can I have the slides? So I'm working actually in France. I'm working in Versailles actually, and just beside the palace. And you will be warmly welcome to come and visit. And we are actually been involved in breeding for many years. Actually, Versailles, you have to know that for the Louis XIV, it was the earliest place where breeding was taking place. And you can have a nice, very nice vegetable garden that you can visit and see what breeding can do. So breeding is something that in plants occurred for a very long time. Yeah, please. Okay. Yeah, sure. And well, in 5-10 minutes, but probably more than 10 than 5, I just would like to review a little bit of the current status of CRISPR in plants. It's been really a revolution, not technically, but with what you can achieve from it. And then I would just give you one little story from the lab for you to see what we can achieve, and maybe change a little bit the paradigms in terms of breeding and agriculture. So the first thing you heard about it, CRISPR is basically, it is simple. I mean, for biologists, it is simple. I've been doing GM crops for almost 25 years, and that made my life much, 100 times more easy. I won't go into details in the little scheme, but there are two things you have to know about the CRISPR. You have two requirements. The first one, which it seems obvious, but it's not easy, you have to know the target. If you don't know the target, it will never work. You have to understand that breeding has been working for thousands of years without knowing what the breeders were working with. We're not knowing the genes, they were just knowing the plant they were growing in the field. However, for CRISPR, it's a biotech tool, you really need to know, that's the first step in the design, the little orange thing, you need to know what you're targeting. So that involves all the genomics behind that, and all the techniques of the genome knowledge and annotation. The second thing you have to achieve with the CRISPR is to have to get this little piece of DNA, or actually RNA also, in the cell. And for some plants it's easy, for some other plants it is not. So with these two requirements, usually you can get your plant. So the major breakthrough is that it's quick, very cheap, and relatively easy. I didn't say easy, but it's relatively easy. The second very positive thing, it allows access to many traits, for instance, known in one species that you can translate into another one. That's we call it translational research. That's very important. You can move from your model plant to a crop plant very easily, because in the model plant you know the target, and then you can predict that it will be the same target in the crop plant. So that's a positive side. On the negative side, as I told you, it requires plant transformation. You have to get this little RNA and nuclease into the cell, or actually the gene coding for these elements into the cell. And the second one, and I will just come up a little bit, even though it's not the topic of today's, it's the regulation. Is it a GM or not? Is it genetically modified or not? And you will see with my little story, that's something that's really it's not science, we're not talking science anymore, but we're talking regulation. So basically in plants it has exploded like in other organisms, and I just took it from a recent review. Basically the first plant is a model plant, but all the other plants you see on the right are actually crops plants. And you can see that with the years, every kind of plant has been actually CRISPRized, if you want. The second thing is, what are we doing CRISPR for? We are doing CRISPR for the obvious classical traits. The first one is yield, obviously, the classical excuse that we have to feed the world, which is still true and will be true. But also you have to see that the other traits that start to be very interesting are actually resistance to stresses, and we are here moving into more sustainable agriculture, and we'll come back to that later. So that's for the overview. Now we'll try to move into my little story. So we're going to focus on one trait. This trait is fat. Fat is really good. It's very good. It's actually essential. You have to know that 80% of fat in the world is actually coming from plants. And more we will need the vegetable oil. And as you can see, the demand for vegetable oil is increasing. What you do see as well on the curve that you see on the bottom, there are mostly 4 plants that provide the oil in the world: palm, rapeseed, soybean, sunflower. And all the rest are minor producer in terms of tons of production. And it's mostly used for food and feed on one side, but also used now for biofuels, but also for industrial feedstock, and obviously to replace, like actually our ancestors that were using oil for the lamps or for burning and heating, we still need oil for that. What you have to know is we don't need we do need many types of oil, and oil has some specificity, and I would just stress on one. This one is what we call oleic oil. It's the one coming from olive oil, obviously, but actually used now in industry and from many other sources than olive. Olive is actually very expensive to produce. So now, actually all the oleic oil that you can find on the market for the agro-food is actually coming from rapeseed, soybean, sunflower, the frill, and very little from olive oil. And the market is increasing dramatically. Why? Because olive oil is considered, or olive oil are considered as very good nutritionally, very stable. And it has to be, and it will actually replace saturated fat in the food, in the processed food. So that's a very important thing. So now you have to produce more olive oil. So how do you achieve that? So there are two ways. The first strategy, the classic one, I will say, is just breed the cash crop. The cash crop is the big ones, the 4 ones I told you about. And here's the very good example from Calyx, a US company, that's actually edited. It's actually not CRISPR, but it's the same principle, edited soybean to make olive oil. And you have the profile of the oil with a little bar, and the pink bar is actually the oleic part of the oil. The plant got the regulatory clearance in 2015, and it's been started to grow in the US, and the commercial launch it was commercially launched last year, and I think it's on the market this year. So that's successful, but still, it doesn't actually answer all the problems. And one of the problems you're going to have is that if you have more crops like that on the same arable land, it will compete for all the others, with all the others. So one way to turn around the problem and say, well, can we grow plants on different areas? And you can think of many different areas or different way of different cropping system. And one way is to look for what we call marginal land, the poor lands. The one you will never grow cash crop on it because it will cost too much. So why not we use this land, there are no forests, so we're not talking about deforestation, but it will be ready to grow, but you have to find the right crop to grow on this land. And that's where my little story come in, is that we actually use CRISPR to work on the forgotten crop. This forgotten crop has been grown for thousands of years in Europe. You can find it in sites in the Middle Age, for the Roman times, etc., all across Europe. It's called camelina. It's a little brassica, very, very tolerant and robust. It's a low input crop. You can find winter, spring cultivar. It grows quickly. The yield is not very good. Obviously, it has never been bred, so it's like an old crop that's just coming back. What's interesting, it's very high in omega-3, which, very interesting nutritionally, and been authorized for feed and food. You can find it, for instance, in France, we can find it in organic shops. And it has a lot of interest in mixing with other crops. Russia has been growing this crop for a very long time, and still growing it. And in France, mostly the organic sector. So what can we do? Can we actually breed this plant? But we did it. The idea was, can we convert this plant as an oleic-producing crop? And we did it. And what we did is we targeted actually free genes. Like wheat, we have free genomes in the plant. So it's something that's not achievable by classical breeding. The odds of getting free mutations in the 3 different genes are almost non-existent. You will have to grow billions of plants to get it. So we targeted these 3 genes, and we obtained what you see. All these little blue bars are individually align a plant. And these plants are actually scored with the quantity of oleic acid you have in the oil. And you can get all different classes of plants. And that's great because you can it's like going to the supermarket, say, okay, I want the 20, I want the 30% content of oleic acid. Just you pick the crop you want. And the little red and purple bars you see below are the mutations that have been induced by CRISPR. So as you can see, more mutations you have, the more bars, the more colors you have on the right part of the graph, more oleic acid you produce. So the fantastic things of CRISPR is exactly that. You can get increased diversity in the system, and you can create diversity you could not find into nature. We tried, and we failed. So the only way to retrieve oleic plants were actually by CRISPR. So that was a fantastic achievement for us, and I'm very proud of it. Well, that's a lab thing. The real life is out in the field, and that's where the trouble came. So CRISPR is easy, regulation is tough. So last year, we asked the UK government whether we can grow this plant in UK. And the DEFRA, the UK, let's call it the Department of Rural Affairs, agreed and said, yes, these plants are not GM, no problem, because they have no foreign piece of DNA. Mutation could have occurred naturally, the same mutation, so yeah, go ahead. So we grew them. So that's with my friend Jonathan Napier from Roslin Institute, north of London, in the field, and say, well, we went and grow. And it was very interesting, the result that they came out was very, very nice. Unfortunately, in the middle of the field trial, the plant changed status. They became GM by the decision of the European Court of Justice that probably everyone knows. And in the middle of the trial, we had to change the fate of the plant because they were GM this time. Fortunately, these plants were grown in a GM condition. We were actually cautious, and we knew that it could happen, even though we didn't want it to happen. So here you have actually the field in the Roslin experimental station. And what I can say, and I will leave my story here, is that we are continuing field trials. I think we were the first one in Europe. Another one is actually running this year. I've been in London last week to look at the plants, and they're looking very nice. So one of the issues we have, and we really made a statement with Jonathan with a recent paper that's quoted here. For us, it's essential that we have access to the field. The field is the ultimate experiment. You cannot predict whether a variety would actually perform if you don't grow into the field. So at least for research, we're not arguing that we should have all these edited plants in the field of Europe. While it's not regulated the same way in other parts of the world, it's important that European research can still go to the field. And that's where I will leave, and I will be open to any questions on that matter. Thank you for your attention.
Thank you very much. Chris? We'll get to questions after Chris's talk.
Okay, my name is Chris Proudfoot, and I work at the Roslin Institute at the University of Edinburgh, and I've been asked to talk to you today about our efforts to edit the future of livestock species. Obviously, John's given us a very nice introduction to the CRISPR-Cas system, so I will skip that and go straight to our use of genome editing in livestock. At Roslin, for the changes that we are wanting to introduce, we prefer a direct approach where we simply inject the reagents directly into the zygote of livestock species, that is the single-celled embryo. And these are then transferred to recipient animals to hopefully result in edited offspring. We've had a number of successes using this technique in pigs, sheep, and cattle. Obviously, at this point, a question we often get is, why would you want to use genome editors in livestock? And there are a number of answers to that question. And it basically revolves around variation or traits. You can introduce novel variation to a breed. You can move variation between breeds or species. And you can also eliminate deleterious alleles. And the flip side of that is you can actually introduce deleterious alleles, which seems slightly counterintuitive, but it's particularly important if you're looking at generating models of human disease. And that's where I'll start. So animal models are very important for science, but the big issue is translational efficacy. You've got to make sure when you're generating an animal model of a human disease, you've got to make sure that you're using the right model that will give you the right answers to the questions you're asking. If you're looking at a neurological disease, if you look at the gross anatomy of the rodent species, often used as models of human disease, you can see that from a gross anatomical point of view, There are various different structures, smooth compared to the Juranocephalic human brain, and histologically the ratio of grey matter to white matter is very different from humans. If you look at sheep, on the other hand, you can see that structurally they're very similar to humans, and the ratio of grey matter to white matter is very much closer to that is observed in humans. And this is why we use sheep to generate a model of Batten's disease. Batten's disease is one of a family of neurodegenerative disorders, and there are 14 different forms caused by different mutations in different genes. And generally, the variation is to do with the age of onset and the rate of progression of the disease pathology. But generally, the pathology is sort of very similar with the various mutations. So we decided to look at generating a model of PPT1, which is the earliest and most aggressive of these diseases. It's the most frequent form of dementia in childhood, and classic symptoms are blindness, cognitive motor deficits, seizures leading to irreversible coma, and unfortunately patients suffering this disease would be extremely lucky to see their 10th birthday. And also a classic sign of this disease in humans is the presence of this autofluorescent storage material, which if you look at the picture, which is a cross-section of a brain, you can clearly see a massive accumulation of this storage material. So to generate the model of Batten's disease in sheep, we used a CRISPR-Cas system to introduce the human mutation into the sheep gene. Essentially, it's just a stop codon, so it prevents a full-length functional protein forming. And we generated a number of animals using this technique. And here are a picture of some of them here. So the question we have at this point is, have we generated an accurate disease model? So if you look at the blood, we can carry out an enzyme activity assay, and we can clearly see that the homozygote animals have very much reduced PPT1 enzyme activity as we'd expect. We can carry out clinical scoring, such as the menace reflex to assess cognitive and motor deficits, mobility and gait balance, which, much to the amusement of the farm staff, involved spray painting the feet of the sheep and getting it to walk along wallpaper. Unfortunately, one of the sheep decided to hop or skip along the paper, and when we analysed the gait, it looked like it had missed a leg. We can also carry out non-invasive imaging, and this is guys using a camera to look at the vasculature at the back of that eye to see any differences. But at end stage, we're also able to look at the spinal column to look for these aggregates, and you can clearly see that they're present in the affected animals and not in the wild type. In humans, MRI is often used to assess neurodegeneration or cortical degeneration, and we have an example of a patient here between the ages of 2 months and 3 years of age. And we were able to carry out the same sort of analysis using our sheep, with the affected animal showing a reduction in cortex, an increase in ventricle size, and alterations in structure of the hindbrain. All sort of classic symptoms of what you'd see in human patients. And we're also able to carry out volume rendering to actually calculate the significance of these changes, and comparing the ventricle size in affected and wild-type animals, comparing it to the hindbrain, which doesn't change in volume, you can clearly see that the ventricles are much larger in the affected animals. So I apologise for the graphicness of the next image, but I think it's an important one to show. And so this is aged match animals with a wild type on the left and affected animal on the right. So not only does this highlight that we've generated a model that very closely resembles that of the human, but it also shows the devastating effect that this disease has on human patients. age-matched animals with a 40% reduction in brain mass. So at this point, we now feel we've successfully generated a sheep model of Batten's disease, and we're looking at carrying out a trial for an intervention study to look at validating a gene therapy study, a gene therapy method for human patients. The second example I'm going to talk about is porcine reproductive and respiratory syndrome, and this is more of a sort of agricultural one. So the disease is the single biggest disease of pigs worldwide. It results in an abortion in sows, the death of young piglets, and a failure to thrive in older piglets. In the EU alone, it's estimated to cost in the region of 1.5 billion euros annually in losses to the swine production industry. So it's a fairly catastrophic disease. The disease is caused by the PRRS virus, and it infects macrophages, and it does so through specific interaction with a cell surface specific protein known as CD163, and specifically domain 5 of that protein. So if we look at the gene structure in the genome, domain 5 is encoded by a single exon in that gene. So it gave us the idea that if we used the CRISPR-Cas reagents to simply cut out that region from the genome, would we generate a protein that was still expressed, still in the surface of the macrophage, and critically still retained the functions associated with the remaining domains, but most critically would no longer act as a viral entry method. So we generated a cohort of animals that had this mutation, and here's the 8 here. So we have 4 edited animals and 4 wild-type animals. You can't tell the difference between them. They're all happy, healthy, thriving piglets. They grow as you'd expect in a normal farming environment. But critically, if you inject these animals with the PRRS virus, the control animals are infected as you'd expect, whereas the edited animals are totally resistant to the virus. And this, to me, is a perfect example to highlight the power of the CRISPR-Cas system. PRRS is the biggest disease of pigs worldwide, and in a single step, we've generated a cohort of animals that, when directly injected with a virus, are totally resistant to it. That, to me, is quite astounding. So in terms of the future, really, I think the possibilities are expanding. The technology is moving at such a rapid rate. Our ability to target multiple traits, or multiple genes, multiple target sites in animals is being investigated. We're sort of limited more to a single target at the moment. The technology, as the 2 speakers before me, is developing at such a rapid rate. I really do believe that we're in a really exciting time in science. With that, I'd thank you for your attention. I'm happy to take any questions.
Thank you, Chris. So one of the things that I think we want as journalists is story ideas, and I hope that we can toss out some ideas and you can ask some questions to sort of hone in on that. One of the things that I also think is difficult about CRISPR is covering CRISPR is there's so much hope here of radically altering the world for the better. I mean, it's not hyperbole to say you could get rid of this disease in pigs, or you could make a crop that doesn't exist right now, that doesn't grow in much of the world, to supply a tremendous product that we all use, oil. But what's the distance between your scientific accomplishment and moving that forward so that people actually benefit from it? And what are the scientific obstacles that remain in the way? Even though you've shown in a small study, for example, you showed the pig brain in Batten's, but the real question is, can you then use that model to develop a treatment that can help humans? And what's the distance between where you're at today and this actually helping humans? What's the distance between that crop that you have shown can grow with the edit you've introduced, and humans benefiting from it? What's the distance between the hope and the hype? You can have your own mic also.
I think there's a number of different challenges. As scientists, taking the PERS model as an example, we've got to carry out our due diligence and make sure that the model is sound. We want to make sure that we haven't had any unintended effects to the animals. We haven't inadvertently made them more susceptible to more run-of-the-mill diseases. Carrying out co-infection studies to make sure that there's no negative effects.
You're referring to the porcine
Yeah.
respiratory.
Yes.
And several labs have shown that, right? Your lab is one of, I've read
three.
at least 2 other studies,
Sure.
right?
And so I think as scientists, that's the important next step for us. The more difficult one, I think, is actually getting it from a science lab to the bacon in your sandwich. That's going to be a tricky one, and it's a debate that's going to be ongoing for a while, I think.
For regulatory reasons.
Basically, yeah. I think crops in the US certainly aren't under such strict regulation. I think in the US, genome-edited animals are regulated as a new drug, which does make
They are. This is an interesting distinction. I think this is important too. The USDA regulates crops, agricultural crops, and they issued something called the mushroom letter. If you've never seen the mushroom letter, you can Google it, it's worth reading. But the mushroom letter basically says that if this could have happened naturally, and this was about changing mushrooms with CRISPR so that they don't brown as quickly. If this could have happened naturally, if you're not introducing any new species, DNA from a new species, then it need not receive any regulation from USDA. So that's the mushroom letter idea. And that letter has been written about many other crops. But when it comes to animals, it's an FDA decision. And the FDA says that if there's one change made to the genome that's intentionally made by a scientist, then it's just like a new drug, and it has to be regulated. So the FDA doesn't share the same perspective with animals as the USDA does with crops, and many countries look to the USDA for good or for bad, or look to the FDA for decisions about how they're going to proceed. So anyway, that was a long aside, but go ahead, Devin.
Yeah, so I think that's an important discussion, but I think we need to persuade the regulators to. I mean, we're very happy with regulation. I think regulation is a good thing, but we want regulation based on the risk and the actual scientific risk, as opposed to the fears.
Jean-Denis, do you have a mic? Yeah, we were talking about this earlier. Naturally, plants and animals mutate, right? How much mutation occurs naturally? And that's never regulated, right?
No. So it has been looked at carefully, and it's roughly 1/10 to the ninth. So roughly, with the size of the plant genome, you can expect like every seed is different at the genome level. So when CRISPR is you differentiate like to one nucleotide, I mean, plants will mutate at the same rate, basically. That's why, for instance, we have all this debate about whether CRISPR could induce some other mutations that would be a problem. Even though in human, that's really an issue. While in plants, it is actually not, because the plant mutates naturally at so high rate. For evolutionary reason, plants have to adapt. So in one way, since the plants are sessile, they don't move, one way to adapt is to mutate. And so you've been eating mutants at lunch this morning, yesterday, all your life. All around us are mutants. And so what CRISPR is doing is actually increasing, or more specifically, will target more specific place of the genome to get the mutation you want. So breeding has been just a way of selecting what nature has actually, the diversity that nature has been produced. was produced. Sorry.
So I'm going to suggest that this is a good story idea. To look at the natural mutation rate of different plants and animals and compare it to what's being done in laboratories. Because what's happening naturally dwarfs what's being done in the laboratory, right? I mean, the mutations you're making, in a pig, when you make an embryo, how many mutations naturally occur? When they fertilize naturally, forget about in vitro fertilization.
Yeah,
do you have an idea?
In the region of thousands.
Thousands of mutations.
Yeah.
Yeah.
So thousands of small nucleotide polymorphisms, small changes. Every pig is slightly different.
And we eat that meat without regulation.
Yeah.
There's no concern of humans being harmed by regulatory agencies. So to me, that's an interesting story. I think the engine of all good stories is conflict, ideas hitting each other. That's an idea that, to me, is fascinating.
Well, the thing is, if you go to South America, there's not an issue with this. South America.
What do you mean non-issue?
Well, Argentina and Brazil, as far as I'm aware, if you're deleting something from the genome, it's not an issue.
With animals as well?
Yep. Which is why I believe genus are looking at taking this down to South America.
Who is that you referred to?
Genus.
Oh, Genus, the company.
Genus, the pig breeding company.
So it's a UK company that also has outlets in other countries.
Yes.
Right? Yeah. Why don't we open it up for questions as well? I mean, I can pepper them with questions all day, but maybe we can pass the microphone around so you can hear.
Thank you. My name is Philippe Morillon. I'm coming from the field of microbiology and antimicrobial resistance. I like the debate about the mutation, but there is something missing. It's the phenotype. And I give you just examples. If you ask a bacterium to mutate for antibacterial resistance, it would do very quickly, or it would get outside DNA and become resistant. Now, the resistant bugs is less fit, is more fit in the presence of antibiotic than the other competitor because they are not resistant. But if you remove the antibiotic from the medium or in nature, it is less fit than the natural, the one that was before. It would disappear. So one thing I was asking, I was wondering, when you make a mutation in plants, the mutation certainly exists naturally in the population of plants. Now, you could select from the natural occurring mutation your plant. Maybe it's difficult because you don't have the means to select it, because maybe it's less fit. I don't know. So what is the phenotype of these bugs? Because among the zillion of mutations that you have in the real life, there are at least billions that are lethal. So you can play lethal in certain conditions, being outside temperature, whatever. So the question is basically, do you look at what are the constraints of the environment that allow now this plant to grow adequately?
Yeah, it's actually a good question. I used to tell my students that if you will grow cauliflower in the field, do you think you will get cauliflower the next year? And you won't. A cauliflower is a fantastic double mutant that will never survive in the wild. It survives only because man decided it has to survive. So you're absolutely right. A mutation most of the time comes what we call a trade-off. It will come with a negative side. And domestication is basically that. A plant survives in the wild because it can propagate seeds efficiently. That's his way of surviving. To propagate his genes is to propagate the seeds. And definitely a farmer absolutely don't want these plants to propagate seeds. So actually the first step of domestication was actually to keep the seed on the fruit. That for wheat, that's the best example. So you're absolutely right. So that's why I think we need to go to the field. Why? Because the field is a place where it's a very stressful place for a plant. I mean, it suffers drought, heat temperature, cold, pests. And obviously you might have a fantastic plant in your lab, just grow like a fantastic yield, and then you put it in the field, boom, it's dead. because there is this pest that you didn't think about, or you just lost the trait that helped the plant to survive. And so what I mean is that I think we need to have these field trials to know whether we can select for the right trait. And I would go one step further, as I think the miracle plant or the miracle crop doesn't exist. And one of the advocacy we have is we should diversify our resources, our genetic resources, so that you can adapt the plant with your soil, your pedoclimatic conditions, your use, and the farmer will have to pick the right one for the right time according to the climate changes, etc. So, and for that you need a very diverse resource and a high diversity in the germplasm. So I don't know if I answered your question, but you're absolutely right.
There's a really interesting experiment that's going on right now with tomatoes, where people have taken wild tomato plants that were never domesticated. There's a collection at UC Davis of these that were collected from South America. And they're putting in genes from domesticated tomatoes because the fitness hit was taken by the wild tomato plants, and they lost traits that are really good. For example, I met a tomato plant that has really sticky leaves. It's a wild tomato plant, and that's an insecticide. And the tomato plants I grow at home don't have sticky leaves at all. So now taking domesticated genes and putting them back into tomatoes is a way to make up for that fitness hit that the wild plant took through domestication. So I'm curious from both of you, and I think these are great stories. Can you point to any other animals? Could we take something from wolves that we lost when the dog was domesticated and put it back into dogs that's a good attribute? Is there something, other plants you can think of other than tomato, where we're doing that reverse engineering?
I think the example I might use would be the polled cattle from recombinetics. Most dairy cattle are born with horns, and it's a major issue due to stocking density and such like. So shortly after birth, the horns are burned off. Most beef cattle, on the other hand, are born without horns, and there's a small genomic change that the beef cattle have that prevent them growing horns. And so in theory, you could take a beef cattle and breed it against a dairy cattle to generate a dairy cow with no horns, but the hit you would take on the breeding value for milk production would take generations, it would take 20 generations to unwind. Whereas recombinetics have taken that small genomic change and put it straight into the dairy cattle. It's born without horns and no hit on the milk production.
I want to push this a little further, though. Is there something wild, like a wild boar, that's never been domesticated, that has an attribute, has a trait that you might put into pigs? Have you thought about that? Any animal that has a wild type, that has a trait that got lost through domestication?
Well, our group's worked on African swine fever virus. So in sub-Saharan Africa, warthogs show persistent signs of infection, yet no clinical symptoms. Domestic pigs die of a highly hemorrhagic fever very shortly after infection. And so we've looked at differences between domestic pigs and warthogs to see where the changes are. And the REL-A gene is one gene that we've moved, which REL-A is one half of a transcription factor which regulates immune response. And so our theory was that the warthogs have a less active immune response, less active REL-A. And so if we take that into domestic pigs, they would perhaps resist infections. Well, we've done the challenge, and I'm not allowed to discuss the results.
Good question.
My name is Odette. I work for the Swiss Public TV and Radio. I'm wondering, this court ruling that gene-edited plants are now considered GM, how did this affect your work? I mean, besides the fact that you have now to do very confined field trials, do you have any problems getting funding for your work, or what kind of effects did it have?
Oh yeah, I can give you a very nice anecdote about that. Last year, we coordinated a European program to actually breed camelina to be even more resistant to biotic stress, to drought specifically. And I included some gene editing in the program. And each of the partners had to check with their legal authorities whether it would be fine, and everything was fine. When all this, the project was discussed by members of each countries, our project been rejected, were found non-eligible by Austria. And Austria suddenly decided that gene editing will be GM and will never fund any GM research. And the project has been dropped. And to go further, we're actually setting up a new project for the EU, and we decided to retrieve or not to include any GM or GE plants, because we think that will be rejected by the EU panels. So yeah, it's affecting a lot. So it's even more cumbersome to do these trials, because these trials are very small. I mean, well, I didn't say, but it's probably 1.5 meters up to 2 meters. So it's nothing like the real crop system. It's cumbersome, but also I can only grow a few lines at a time. And so for many of us, it's a very strong limitation, but again, because it will restrict research to the lab. And we cannot do this last leap forward by testing the lines in the field. I mean, I totally accept that society doesn't want it, a European society. I do accept it. The only thing, it takes 10 years to create a new variety. 10 years. It's even longer for trees, obviously. And let's say you have an outbreak of something really nasty. It takes 10 years. So if we don't have it in the drawers, I mean, it will take time. So I really try to convince people and say, well, let's at least do the research in the field and maybe keep it aside because you don't want it now, but at least we do have the resources in case we need it.
If scientists are right and GM with CRISPR and other gene editing technologies is beneficial to humans, do you envision a day where we will label things GMO instead of non-GMO and that people will seek out those products?
Difficult one to say.
Would you buy a product that said GMO as opposed to non-GMO?
I'm not sure I would be particularly bothered one with the other. The GM salmon is being consumed in Canada at the moment. I think in the US they're still arguing as to how the product's labelled, but it may be a while, considering it took 29 years for the Aqua Advantage salmon to be approved. So we may be having this discussion for some time.
Other questions? Yeah. I think it would be very cool for a company to put out a GM label on their food.
Good comment, yeah.
I'm kind of Helen Gavigan, Science People and Politics. This European Court of Justice ruling, first of all, at some point before I leave, I'd like to get the case number. I'm curious about their reasoning. Isn't there an essential philosophical difference between breeding and mutation? And I just want to plant that there because I've not thought about it, whether philosophically, essentially, mutation and breeding are fundamentally different. But one of the things you said so regulation is fine, surely. I mean, if you can go ahead and do the experiments. So how can we be certain, or how can you be certain that all the possible changes would and could arise by mutation? Isn't that... Do we know enough about the biochemistry to be able to say that? Irrespective, and I think all of the things you said were absolutely fascinating about comparing the number of mutations naturally arising and all the rest of it. But what fundamentally about the biochemistry do we not know that might undermine us being able to say absolutely all mutations could arise, therefore it is a naturally occurring product as opposed to one that's genetically modified?
Okay, I'll try to answer that. So it's a real, very important question. What the European Court of Justice has ruled that actually the process, it does not rule the product. That's what makes a big difference with Canada or US. In Europe, whatever is happening, if it's the process evolved at some step, the introduction of foreign DNA, and it did in my case, it is GM, it will be GM for all the progeny, even though it's the grandparents of the plant, even though I've proven that there is no piece of DNA left. So now the question arises. I can demonstrate that there is a mutation, the mutation I want is there. That's actually what I produced to the DFRA, the UK government. I produced look, that's the changes I've made. I also provided the fact that there is no other piece of DNA in the genome. And the UK government has only that as a proof. On the side, I sequenced my lines. So I know all the mutations in my plants, all of them. And believe me, there are plenty. But there are as many as my wild-type control. So I don't even know whether these mutations are coming from. So honestly, these mutations are there. All my plants are different, basically, except for the 2 mutations I've introduced. They are all the different mutations. So I'm stuck there because I cannot tell you and affirm that indeed these mutations are coming from CRISPR or not. As I told you at the beginning, plants mutate a lot. So what we do know, there's no higher incidence of mutations. That's the only thing I can tell you. Now, whether it's dangerous or not, I can tell. What I can tell you is that the mutations I introduced, they are not dangerous. I mean, you have probably eaten sunflower oil from the same mutants. The only way, but you're right, maybe some of these other mutations could have produced a negative trait, and there are negative traits. I mean, plants produce some toxins, and I mean, that's for sure. So we don't have to turn around. I mean, that's true. So the only way is to grow them, like breeders do for thousands of years, and just see whether you have a negative trait like you asked last time, or whether it's a real positive trait. So the only way eventually to select what you want is the phenotype, so how it looks like, and etc. But fundamentally, the plants are not very different from their counterparts that's been selected naturally.
When breeders select, they're selecting in context, aren't they? So they're selecting in an ecological and environmental context for what is going to work. So that's breeding as opposed to mutation. And I'm not even talking about dangerous mutations, whatever that might be. It's really this very fundamental, before you went to DEFRA, of saying that, what is it you said? Just let me find out what you said. Because I thought, how can you be certain that what you're looking at could have arisen by mutation? What I'm really trying to say is there not, can you be 100% certain of the fundamental biochemistry, evolutionary context, that argument would hold up? Now, I haven't read the ECJ judgment yet, but I'm guessing they think not. Maybe somebody should translate the question. I'm not sure I'm doing it, putting it terribly well.
Yes, the mutation we've introduced are different from the one from soybean, sunflower, and rapeseed. So, contextually speaking, they are different. However, in terms of biochemistry, we know exactly what we've been doing and what this enzyme is coding for, and so that's for sure. We know exactly what happened. But you're right. The breeding process is not what happened in my lab. The breeding process is what's going to happen in the field. And what we just ask for is to do the breeding, is to do what breeders do, which are growing the plants for years and years and check that everything is fine.
The case number is C528/16, and it was July 25th, 2018.
Can you give me that one again?
Yeah, it's C528/16 from July 25th, 2018. I'd flip that question on its head.
I want to rephrase the question. So if we waited for 1 million years or 1 billion years, this mutation that you introduced would have happened by chance anyway?
Yes.
To flip it on its head, though, I wonder, when breeders breed new plants, is there 100% assurance that that plant is safe? I'd say no, that scientifically you would put it out, and if a problem came up, you would pull the plant. That's my guess of how it works.
You're correct.
And so I'm not sure that this is any different. You never have 100% certainty of anything, and problems occur. So scientifically, what's the dividing line? That's where I return to.
I'm not convinced there is. It was the line about 100% mutation, rephrased by this gentleman here.
Yeah.
to help save all of us from my fumbling around. Yeah.
Other questions?
Then maybe I have a question of my own. Don't you fear that your good intentions will end up at something like Monsanto locking us in a special business model?
Okay, we're shifting something else. But it's back to one of the questions. What is fantastic with CRISPR is that we might change the paradigm of breeding with the GM. Right now, the GM has been actually developed only by the big agro system for monoculture and a very specific cropping system. And most of the benefit actually doesn't go to the consumer. It will go to the breeders, to the herbicide producer, and the farmer, but not the consumer. The problem is so expensive to actually make a GM that actually no one else but Monsanto and Syngenta could do it. Well, with the CRISPR, since it's cheap, the discussion I have with breeders is, well, maybe now we can develop things for the consumer. I will take just an example. I discussed with the wheat breeder in France, and I was discussing about gluten-free wheat. They said, no way, it's a niche market. We never go there, it's too expensive. But a small breeder might say, well, if it's cheap and easy to make a gluten-free wheat, why not? And that's who's going to benefit from that? That's a consumer. So maybe now think about differently. You can say, well, maybe we can think different traits now. We can think traits that would be agroecology, for instance. So you will grow differently. There will be maybe niche market, but since it will not have cost so much, I mean, it will be profitable still. So I think now we can maybe change it. That's why, I mean, putting these plants into the GM burden in terms of regulation, would at least on the European side, would just kill this initiative, for sure.
Chris, do you see niche livestock the way that Jean-Denis is describing for plants?
Not so much in
Bama pigs are very popular in China. These miniature pigs, people love the flavor of the meat. You know, can you see something like that?
Yeah, but potentially. Sheep breeds very individualistic, very much smaller scale. In terms of the pigs that we work with at the moment, it's mainly sort of large white landrace, which is a very sort of generic,
well, I know
bog standard pig.
I know you don't do this, but one niche market for CRISPR with animals is pets, and introducing traits in pets that you want a Dalmatian with purple spots. You know, these things are possible now.
In theory, yes. Whether
I think it's likely going to happen.
No.
Why not?
People have. I often find that when you have a sort of ethical debate about using sheep as a biomedical model and then a dog as a biomedical model, I think people see dogs as companion animals.
I'm not talking about biomedical model. I'm talking about the eccentricities of pet owners who want certain traits in their pets. I mean, look at our variety in dogs. Those didn't occur naturally.
Or the glowfish.
Yes. So that's what I'm suggesting. Could that be, given the ease and inexpensive aspects of CRISPR, could there be an animal market for pets that's niche for Dalmatians with purple spots?
I think if somebody's crazy enough to take it on board, then possibly yes. I mean, the glowfish seem to be quite popular in the States.
Thank you for pointing out the States and looking at
Sorry.
That's how I did it.
Well, I showed the picture to my kids and said, "Oh, Daddy, can we get one?" I said, "No, we're not allowed to buy them in this country."
You can't buy goldfish in
glowfish. Glowfish, the glow-in-the-dark fish.
Oh, the glow-in-the-dark fish. Okay, sorry, it was accent problem. Other questions? We're running out of time. Yeah.
Hi. Yeah, I actually have two questions. The first being just talking about the different kinds of GM, where you're talking about introducing foreign, I guess, properties that are outside of the strain. And then you're talking about reintroducing certain lost properties, like, for example, with the wild tomatoes. Could that open up a way to, like, classify the different types of GM? So, like, ones that are lower risk, I guess, as understood by consumers, and higher risk, for example, when you hear about Monsanto injecting food varieties with fish properties or something like that. Would that be a possibility? And do you think that would open up more receptibility from consumers, from policymakers, and the like?
So you're right. So there is something behind the CRISPR which makes things a little bit more complicated, is that you have three types of CRISPRs, basically, or the product. One is what we've been talking about, is a mutation that actually the plant or the pig is actually making. So basically, the CRISPR is a scissor, it cuts, the cell repair, and once in a while, it makes a mistake. So that's actually the plant or the pig who did the mutation, not us. And we have basically not the control of the mutation, the type of the mutation. We know where it is going to happen, but not the control of the type. So that's the first class. That would be probably the easiest because that's the one that could occur naturally, because it's actually the plant who did it naturally. And the plant has problems of repairing its DNA all the time. So that's something which happens all the time. That happens right now in every cell of us right now. So that's something natural. On the other side of the spectrum, you could cut and provide, it was in one of John's slide, I think, and you can provide a template. So you trick the cell, and the cell, instead of taking the template of one of these other chromosome, it will take your template and will repair with your template. So you introduce a new mutation, and this new mutation could be a foreign piece of DNA. That's clearly a GM. So it's a CRISPR, that you made a CRISPR with a GM with a CRISPR. And in the middle, you can introduce an allele, so it means a specific mutation that will provide, for instance, herbicide resistance. So it's a very, very specific one. So nature can actually, so it will depend from the type of mutation. Some mutation will be difficult for the nature to make, or will take billions of years or to fill the whole earth with your plant. But some of them could actually occur naturally. So that's a gray zone. We don't know whether it's GM or not. The other one we clearly, it's clearly different. So I don't know if I'm clear, but so basically the mutation that actually the organism is actually making is probably the one which is the closest to what's occurring naturally. And in this case, again, it could be a beneficial trait or could be a negative trait. It will very much depend what target you're looking at. And again, I'm not saying that the process itself could be bad if you just target a negative trait. I don't know if I answered your question. I'm not sure.
I think that's the problem with this whole discussion is that there is such a grey area. There's not one type or the other. There's so many different changes that you can generate from very small changes right up to inserting whole transgenes, and it's where do you draw the line at that point as to everything this side is good and then everything that side is bad, and everybody's going to have a different place where they put that line, and I think that's the problem. But I think ultimately, I think if we look at the resulting product and assess that on its merits as opposed to how it was generated being irrelevant, and actually assess the crop or the pig on its merits, I think it's probably a more sensible way to go.
I guess what I'm getting at is a problem for consumers or even just for journalists writing about this is the understanding. I mean, how do you, without any prior knowledge, how do you assess that pig on its merits or that modified grain? So I guess just see if is there a way that a new language could be developed around genetic engineering that could be better understood by consumers and by those outside of the industry. I mean, at the moment, the discourse is dominated by, I mean, as soon as you think of GM, even if you don't know anything about any of this, you know, you go straight to Monsanto, you go straight to understanding different theories about how negative it is. So
I'm going to put this back on us. I think you raise a really important point, and I think it's the role of journalists to clarify it. And it's a good story. And we probably should end on that note because we're out of time, but I think there's a really good story for you to do and for all of us to do about clarifying that, because it does matter. So thank you all for attending.